Okay. Mira, se me fue el volumen. I cannot hear him.
Hello.
Ashley,
2 years. So technically, there's kind of 6 time points. This is only looking at 3 of those distinct ones. So this is the overwintering. So this is our very first collection from 2022. And then we have bloom and harvest from 2023 that we've looked at. Now, we only started with those because it's a little bit time constrictive, and this is just what we've processed so far. We were able to screen 313 isolates out of that. Each of those were tested twice. So again, that's 606, 26 evaluations we've had to do, looking at every well and doing comparisons there. So the data, this is looking at the tolerance across time. So what this chart is showing you, so on the y-axis here, we have frequency of fungicide tolerance, and then across the x-axis, we have our different fungicides broken into FRAC 9 group and FRAC 7s. And then for each of those bars, they're color-coded by the time point they were in, whether it's overwintering, which is represented in blue, bloom in green, and of course in raspberry color at harvest. And then each of those numbers associated at the bottom legend there are the amount of isolates that we tested in those and screened. So these are percentages. These aren't actually numbers of isolates. But what you can see, I guess the takeaway message I want you to see from this data set is that we're seeing a reservoir of resistance that's overwintering. In other words, for every botrytis we tested from raspberry tissue, we were seeing resistance in. So for example, just take fluopyram. We're seeing 70% to 80% over almost an entire year period. It's consistent. So there seems to be a reservoir of resistance that just seems to persist from season, from year, from mid to end to beginning of next year. Another trend we're also seeing is that the more chemicals we looked at and the more isolates we looked at, we're starting to see resistance that's popping up in chemicals that aren't even sprayed. So fluxypyroxate's here. I mentioned it's not even really being sprayed in the fields much. However, we're seeing resistance that's popping up into it that almost equates to what we saw in boscalid. What this suggests is that we're getting cross resistances in between the FRAC group 7s. So that's kind of an issue we need to look at. And then of course, the last trend that I'll kind of point out, if we want to compare among the FRAC groups, so FRAC 9 to FRAC 7, I know they're different, they have different mechanisms, but we see something like pyromethanol. Pyromethanol is the highest for our FRAC group 9s, and FRAC group 7, we have fluopyram. Lunar tranquility, anybody? Those are the 2 chemicals that are in lunar tranquility. It's kind of a smoking gun for seeing those are the kind of resistance. So based off, our spray programs, those are kind of how we view and see things. And then this is looking at fungicide tolerance, I guess, within a single FRAC group or multiple fungicides. So I know this is a lot of numbers here, but let me just kind of quickly walk through this. This is the exact same data set I just showed you. So y-axis is the different fungicides we tested. The x-axis is frequency of tolerance. And what I want to show here and take away is, so the bottom ones you've seen, the fluopyram, boscalid, fluxypyroxate, these are just the FRAC 7s, not including the FRAC 9s. Those are the same numbers I just showed you on the previous chart. What I didn't show you is the top part here, and this is, I guess, what I want to draw attention to, is multiple resistance for one single isolate that's showing resistance to multiple FRAC 7s. So the biggest takeaway from this would be the isophetamide, the very top one, isophetamide, fluxypyroxate, boscalid, fluopyram. We have anywhere between 4 to 15% of our isolates tested in the same group, showing resistance to all 4 for every single isolate. Now I know previously a lot of FRAC 7, FRAC 9, and even FRAC 17 resistance was documented by Olga and Tobin Peeber's group, probably to most of this very audience in Northwest Washington. The difference here is that we're testing different chemicals. We're not testing the same materials. And what we're seeing is multiple fungicide resistance that's happening within these FRAC groups. I guess the main point, with this particular data set that I want to take away is that we want to maintain our sensitivity to many, if not at least some, of these chemical compounds, even within the FRAC group 7s, so that we continue to use them in the long run. So with that, just some simple takeaways from what I'm having to say. So we have a new assay, spore germination assay. We're continuing to work on it. We're continuing to refine it, continuing to evaluate on it. We're seeing resistance that's developing to some of the newer FRAC 7s, as well as the FRAC 9s that's popping up. And we're seeing resistance in botrytises that are collected from the winter months. So looking at the data sets I showed you, looking at the comparison among the different fungicides, the trend of overwintering and harvest resistance seems like it's going to carry over into next year's crops. That resistance will carry over. It's important for us to identify and screen for this pattern and recognize this pattern, because that's, well, what we can do to predict what might be the best spray application to prevent for next year's crops, next year's season. And then of course, we want to continue our fungicide tolerance monitoring, continue testing our adjustments to our methods to reduce some of the variable results we might be seeing looking at different discriminatory doses, that sort of thing. And we also still need to begin our genetic analysis of our winter botrytis populations compared to those from bloom and fruits to determine interrelatedness. So with that, I want to thank everybody who made this project possible. Washington Raspberry Commission, Washington State Commission on Pesticide Registration, our own USDA, and of course our close collaborators at Washington State University. And most importantly, I want to give a sincere thank you to all of our collaborating commercial red raspberry growers who helped make this project possible. With that, I thank you for your time, and I'd happily take any questions.
Awesome. Thanks, Jeff. I'm going to get this question from Alan first.
Okay.
I have lots of questions, but I'll start with, how did you define resistance?
So we define resistance as anything that was based off germ tube growth, 50% greater in width of the spore, that the germ tube was in that length based on that concentration we were testing.
What makes that resistance?
I wouldn't call it resistance so much as more tolerance. It is tolerant to grow within that particular concentration of that fungicide.
Can you call it resistance if you don't have control failures in the field? Or can you call it tolerance if it still works in the field?
Yes, because we're looking at potential here. When we're calling something, so it's a slippery slope, right? When we're looking at tolerance and resistance, we're testing stuff in vitro, in lab, and what you're actually seeing in the field may be completely different. So one thing that's missing in the field would be environmental factors. If you look at just a simple disease triangle or something like that, environmental factors. These last seasons in harvest have been very, very dry, fairly warm. What are you guys going to do if you get an environmental factor such as heavy rain that might happen at the end of harvest? Right? If we have underlying resistance or disease resistance potential, it could be a bad day.
Is this resistance from use of chemical, or is it from the genetics of the fungi?
It's likely a response of the fungi to use of chemicals. So resistant as it is we're typically selecting by spraying chemicals you are selecting for genotypes or genetic variability within the fungus that's actually, you're leaving those. So you're killing off everything that's sensitive, or you're reducing those populations, and your populations that are left is what has a genetic propensity to be resistant to those chemicals. And there's also mutations that can be happening throughout the season as well that can kind of complicate that factor. So, but that's genetic. So.
Awesome. We got another question back here, and then we'll come back to the front.
Sorry, I walked in a little bit late on this. Would you characterize this resistance or tolerance as qualitative or quantitative?
I'm sorry, can you repeat the question?
Would you characterize this resistance or tolerance as qualitative in nature or quantitative?
I would say this particular assay, we are looking at qualitative, the in-lab vitro assay for resistance. Quantitative, yeah, if you look at and compare it among different frac groups, yes, quantitative in the field. It's growing, especially when you see things like cross resistance, the chemicals that we haven't even been spraying much. So sure, that's kind of cross resistance. But most of these are site-specific fungicides. They're acting directly on a particular mechanism, so they tend to be more qualitative in that nature.
Did you look at the statistical significance on a site-by-site basis? Like, was there any anomalies that were significant from, the mean when it comes to tolerance?
I did not look at that, no. No, these are basically what I'm reporting is basic percentages of what we're actually looking at right now. In generalities, they're not. There doesn't seem to be, just looking at the data on its face, a lot of statistical significance in between some of the different time points or anything like that. But no, we didn't actually go in depth on that.
Does anyone else have any questions for Jeff before we let him go? Awesome. Thank you so much, Jeff.
Thanks, guys.
Okay, so our next speaker is actually coming in remotely. So Molly is going to get him set up. This is going to be Ben Deal. He is an agricultural research technologist in entomology at WSU-Whatcom down in Mount Vernon. And the talk that he's going to be giving is Parasitoid Wasps Attacking Spotted Winged Drosophila, SWD, Surveys from Western Washington. So if you just bear with us, we're going to make sure Ben's up and running and you can hear him all right, and then we'll get going.
Everything look okay?
Looks good here, Ben.
Okay, cool. Well, hello, everybody. So my name is Ben Deal. I'm a scientific assistant now in the WSU-Whatcom lab in Mount Vernon. Sorry I couldn't be there in person. I'm recovering from something that I picked up on my Thanksgiving travel. But I still wanted to talk to you guys about a couple projects that our lab has been working on. These are all focused on this parasitoid wasp complex that is attacking spotted winged Drosophila here in Western Washington. So spotted winged Drosophila, I'm sure everybody is at least familiar with the name by now. You've probably been battling these guys in your fields, SWD for short. So this is the target pest. It's a significant pest in Pacific Northwest fruit production. Other places in the country, all over the world, it's becoming a really serious issue. So particularly in Western Washington, things like berries, so blueberries, raspberries, blackberries, and other fruits like cherries. So the reason that this invasive pest is so big of an issue compared to some other similar flies, the females possess this saw-like ovipositor. So you can see this in the picture on the right. You can see this hardened structure with these teeth on there. It looks kind of like a saw blade. So this actually allows this insect to cut into ripe fruit and lay their eggs. So unlike other flies, like other fruit flies, where they have to wait until the fruit is damaged or kind of rotting before they can attack it, these guys can come in much earlier. They can cut into the fruit, they can lay their eggs, and then you get these maggots in the fruit. So obviously this changes the whole game for growers and any kind of management that you want to apply. So spotted winged Drosophila, IPM, there's a lot of things that are suggested that you can do to try to manage this pest. So things like monitoring for the adults and the larvae. You have cultural controls, so you can clean up all this fruit that's might have eggs or maggots in it, so maybe your population's lower next year. You have things like mechanical controls, chemical controls. So one thing to note here with the chemical controls, there is kind of this idea of a zero tolerance for maggots in especially fresh market fruit. So if you end up having maggots in these berries or something that you're sending to the grocery store, there's a possibility they might get rejected. So this has led to potentially an increase in pesticide usage because obviously growers are worried about this issue and it's their livelihood to sell their fruit. So they're worried about managing this pest more efficiently. So finally, we come to biological controls, which are what we're going to talk about during this presentation. So you have things like natural enemies, some generalist predators, things like lacewings or ladybugs. And then you can also do something like a classical biological control. So what is classical biological control? This is when you bring in enemies from an invasive pest home range. So basically, wherever this invasive pest came from, you can go there, look for enemies, and bring them to the new area where this pest is now active. So here's an example. We have these decapitating flies of imported fire ants. So if you look on the right, luckily we don't have the fire ants up here yet, the Solenopsis invicta, but I was unfortunately lucky enough to deal with them when I was down in Texas, and they are not very friendly. But this is an example where they went back to the home range of the fire ant. They found these little flies, and what these flies do is they will lay their eggs near the head of the ant. The larvae will develop inside, and then once it's ready to emerge, these heads will pop off. You can see the fly emerging from the head here in this bottom picture, and then you have one fewer ant. So hopefully this is something that you can do to manage these pests. So a similar situation happened with spotted winged Drosophila. Researchers went to South Korea, Japan, and China, and they identified about 20 different parasitoid wasps that were attacking SWD larvae. So there are other parasitoid wasps that will attack things like eggs or the pupae, but these ones were specifically for attacking the SWD larvae while they were in the fruit. So from this group, they found 2 species that were identified as kind of the best candidates to bring over as biological control agents. So we have Ganaspis brasiliensis and Leptoplena japonica. Now, obviously, you want to be careful with this process because you are bringing in another non-native species into an area. So this does take multiple years of research in laboratories and quarantine laboratories to kind of figure out the potential impact that this new biological control agent might have. So for things like parasitoid wasps like these, you're doing things like host range studies where you're looking to see what other flies or what other insects these wasps might attack if you bring them in to control SWD. So of these 2 wasps, they determined that Ganaspis brasiliensis was the one wasp that was more specific to SWD, so was the better control candidate. So here's a picture of these the 2 wasps. These are both in the family Phagididae. Ganaspis brasiliensis on the left here and Leptoplena japonica on the right. You can see they look pretty similar. They have these kind of dark, shiny bodies, wings with very little venation, these kind of paler legs and antennae. So they're actually not too bad to tell apart, but we'll see there's going to be a third individual that pops up later that makes it a little more difficult. So while these quarantine studies were going on in laboratories, I think people were just about to start releasing these wasps, or maybe they already did in some parts of the country. Both of these species were discovered in southwestern British Columbia in 2019. And then subsequently in Washington, Northwest Washington and Whatcom County in 2020 and 2021. So somehow these wasps found their way over here. Maybe they had been here for a long time before we knew that. But anyhow, they were already here before people had released them. So that's great news. Now we can just release Ganaspis brasiliensis. Our problems are solved. SWD will be controlled. But unfortunately, with biology and entomology, this is not always an easy case. There's usually something that maybe you might get hung up on. So researchers discovered that there seem to be some differences in Ganaspis brasiliensis and some behavior biology. And they determined that there seem to be multiple strains, so G1 through G5 of Ganaspis brasiliensis. And not all these strains were as specific to SWD as others. So now it turned out that you couldn't just release any Ganaspis brasiliensis. It had to be specifically one of these 5 strains. So this kind of complicated things a little bit. And it brought up the idea of maybe there were cryptic species. So maybe there were multiple species here and not just one. So this is something that we will come back to later on in the talk. So as I said, there was a third wasp that was discovered. So these early surveys, we had these first 2, and then they actually found another species of Leptoplena, Leptoplena heterotoma. This wasp was kind of already in the area. So this is pretty, I think, commonly distributed in kind of the northern hemisphere. So, but this was also found to be parasitizing SWD. But I don't think it's considered to be as effective as the other ones. But telling apart Leptoplena japonica and Leptoplena heterotoma is a little bit more difficult. So that's become an issue in identifying some of the specimens. So these are the 3 projects that our lab has helped with in the past couple of years, looking at these SWD surveys in Western Washington. So first, the Nottingham Berry IPM project, the Abram Island Biogeography, and then Beers Northwest Washington survey. So Dr. Louis Nottingham, professor at WSU-NREC entomology, where I work at Mount Vernon, Washington. So he devised this intensive sampling project to look for arthropod pests and beneficials in berry fields. We looked at both blueberries and raspberries. So Louis is actually going to talk a lot more about this, so I don't want to spoil too much here, but I'll just say that we used a lot of different methods in many different fields to collect and survey these pests and beneficials to see what was out there, and then also kind of when they were active throughout the year. So one of the techniques we did was looking at adult SWDs with these traps here, this picture on the right, which were baited with century lures. And we did this both in Skagit and in Whatcom counties. So as kind of bycatch in these traps, we were getting the SWD adults, but then we were also finding these parasitoid wasps as well. So this year, out of all of our trap samples, we recovered 135 parasitoid wasps from these adult SWD traps. The wasps were collected from traps placed in both blueberries and raspberries, so from both crops. And out of these 135 parasitoid wasps, 94 of the specimens were either Ganaspis or Leptoplena. So this is good news. These are the majority were these wasps that were kind of hoped to be brought in as SWD biological control agents. And of these 94 specimens, 96% were Leptoplena japonica. So this is actually kind of following a similar trend a lot of other researchers have been discussing, where in a lot of these surveys they're doing, the majority of the wasps that they're collecting are actually Leptoplena japonica, which is not the intended consequence here, because Ganaspis is the one that's supposed to be a little more specific towards SWD. So hopefully that ratio will change in the future. And then again, of these 94 specimens, 95% were collected in organic or no spray berry fields. So the next project we helped with last year and also this year, it was repeated, was organized by Dr. Paul Abram in Agassiz, BC. So we had a couple of main research questions for this project. So we found the wasps on the mainland, but they were interested to see, have these parasitoid wasps spread to more remote areas, specifically islands in British Columbia and Washington State. And secondly, if we do find these wasps on some of these islands, could human activity be responsible for the spread? So do we see more wasps on islands where there's more human activity, so more people living there, more ferries coming and going, more goods being moved back and forth, things like that? And finally, on these islands, if we do find the wasps, is there any evidence that we can see for the suppression of SWD populations? So if we find wasps on an island, does it look like they're reducing the number of SWD compared to islands that might not have the wasps? So little methods. We did two sampling trips per season, 45 berries per site. Brought the berries back to the lab to incubate them. After about a week or so, we pulled out all the SWD puparia, and isolated them so that we could make sure the parasitoids that came out came from SWD pupae. We also just left the containers, so we would collect any SWD or parasitoids that emerged from the isolated puparia and also from the container emergence. I'm not going to go too much into the results because this is still kind of ongoing and it hasn't really been published or anything, but at least for us, last year we did not find any parasitoid wasps from the San Juan Islands, so that's where we were doing our sampling in Washington. But this year we actually did recover some from an island. It was from Guaymas Island. So this is just north of Anacortes. It's actually closer than the San Juan Islands. And we have to thank Dr. Charlie Kosler for this. He was doing some other work over there and grabbed some berries for us. And then both years we did find wasps from a couple of our mainland sites. Finally, the last studies, last year, Dr. Betsy Beers from WSU Tree Fruit in Wenatchee. She was part of the team that detected these two species in Whatcom County in 2020 and 2021. So she was interested to see how far these wasps had spread around Western Washington. So myself and Julia Gray at the WSU-NREC entomology, we helped do the sampling. So our survey focused on rearing from Himalayan blackberries, so another invasive species in this kind of complex. And we targeted ones that were growing near blueberry fields in Whatcom, Skagit, Snohomish, and Island counties. So what did we find? We did about 35 different collecting localities. We returned over 7,000 blackberries to the lab. Eventually got pretty smelly in the lab with all these rotting berries, all these molding berries. But it was worth it. We did collect over 700 parasitoid wasps. And again, the majority of these were Leptoplena. We did find some Ganaspis as well, but most of them were Leptoplena. And here's a picture of these isolated SWD puparia in a Petri plate. So additional research questions, things that people are working on. Are releases of these species resulting in established populations. So this is a big thing with biological control. If you're releasing agents, are they actually sticking around? Can you find them later in the season or maybe the next year? Is there any measurable control of SWD populations where these wasps are found or have been released? And finally, the one that's most interesting to me, the taxonomy of Ganaspis brasiliensis. So this is being worked on primarily by Dr. Matt Buffington at the Smithsonian. And we talked about these different strains and if there were any other species hiding there. So it was confirmed yesterday in a meeting that he will be describing a couple new species. So it's most likely that the Ganaspis brasiliensis that we've been dealing with may get a different name here soon. So it looks like there were multiple species there and not just one. So with that, I will thank you and take any questions.
Awesome. Thanks, Ben. Does anyone have any questions for Ben? We can relay them live despite his lack of presence right here. Anybody? Well, in that case, I'm going to give you one more chance. Awesome. Okay. Well, thank you so much, Ben. I'm going to hit up that survey I told you guys about, in which required Thanks, Chris. Awesome. Okay, so this is new to all of us, but we're going to try and get some feedback from y'all on your phones. Because really, honestly, it allows us to improve the conference, but also it gives